Welcome to the GEOTOP-A seminar. This is our first one of the semester. And today's speaker is Natasha Jonoska of the University of South Florida. She'll tell us about topological models for studying DNA self-assembly. Take it away, Natasha.
Thank you very much. And I wish to thank David and Jose and the organizers for inviting me to present my interest and my research. So yes, it's very nice to be part of this long-standing seminar. If you have a question, please let me know. I understand you can put it in a chat or I'll stop, but feel free to interrupt if needed. So I will talk about DNA self-assembly, and in fact, I will talk about the topology that might be of interest to this seminar when these processes are happening. First, I want to mention a couple of words about molecular self-assembly. So this is more or less taken from Wikipedia or something like that. Self-assembly is a spontaneous and reversible organization of molecular units into some ordered structures, but usually this organization happens with non-covalent interactions. So they're not permanent, but usually hydrogen bonds or ionic bonds or some other types of bonds. The formation of the self-assembly system act on a strictly local level, so the molecules interact locally with their neighbors, and we think of it as if the nanostructure built itself. So here I will show a small video, and let me just stop share and start sharing something else. You see my screen? I hope so. So
yes. Utilizing solid printed models.
Can you hear the
Yes.
Yeah, okay.
Of virus components and magnets representing the attractive forces between them, we can produce a physical analog capable of self-assembly by random shaking. The model demonstrates not only how nature accomplishes this feat, but can show some of the subtle characteristics of stochastic assembly. Notice that the subassemblies form and break apart en route to the most stable structure. The strength of the shaking is analogous to the temperature of the molecular environment. If it's too hot, the virus will break apart. If it is in the proper range, assembly will take place at an optimal pace.
So that was the small video that sort of explains how molecular self-assembly works, in particular self-assembly in general. We can think of the temperature and as being a good energy source for the ionic bonds to happen. And that was an example for the buildup of a viral capsid, but I think everything about molecular self-assembly is the whole notion of self-assembly is actually similar to what we just saw. So I want to point out that this notion of self-assembly is something that science has identified. So in 2005, science listed 25 most important questions in science for the next century. And of course, these were the huge questions like what is the universe made of, and what is the biological basis of consciousness? And one of those questions was actually, how far can we push chemical self-assembly? And the concept of self-assembly actually is used increasingly in many disciplines and with different flavors and emphasis in each discipline. From math perspective, when we think about self-assembly, we want to ask the questions about communication between the molecules, what type of molecular communication is involved, what are the shapes that the molecules can actually obtain, and if they're actually performing some kind of computation, what are the results, how these shapes are being built, what type of structures can one make with building block components, and can these structures be identified and described in how, in what ways? What can we actually give some functionality or roles to different types of molecules or different types of building blocks so that we achieve certain structure? And can we describe the whole process? And are there different avenues of self-assembly where we can have the same structure obtained through different pathways? And can we describe this mathematically? So I will talk about DNA self-assembly as a representative of the whole notion of self-assembly. And this is the DNA molecule that many of you might have been familiar with. I just want to point out things that are important in my presentation. So the double-stranded molecule looks like this. It's a double helical structure. It consists of two strands. Each strand has bases, and the bases are these four bases, A, T, C, and G. And they are complementary to each other. C is complementary to G, and T is complementary to A. They make hydrogen bonds. And due to this hydrogen bonding, we actually obtain this double helical structure of the DNA. What's important in my presentation is that the strands are oppositely oriented. When they come together, they actually come in opposite direction. So one end of the strand has a phosphate, and we usually call it a 5' end. And if you move around, and at the other end of the strand, we have a hydroxyl group, and we usually call that a 3' end of the molecule. So in here, this strand will have a phosphate, and that will be a 5' end. And on the other side here, we will have a hydroxyl group, and that will be a 3' end of it. So when the two strands join together, they come in the opposite directions. This is a very small molecule, as we know. It has 2 nanometers in diameter, and one helical turn is about 10.4 nucleotides, 10.4 bases, and 3.5 nanometers. We know the DNA as the molecule of life. So this is a Jacques Rousseau painting where you have bursts of life everywhere, and in all of these species, there is DNA, and that's the molecule that makes us, I guess, makes the basis of biology. But in 1982, Annette Simon, who now is considered as the father of DNA nanotechnology, proposed a holiday junction where he, in some sense, destroyed the symmetry in the nucleotides. Holiday junctions happen during recombination in natural biology environment often. However, they're never stable because they actually have symmetric complementarity here, and the molecules kind of moves around. But if you make it the nucleotides and the bases, if they become asymmetric, then we end up with this 4-arm molecule that is pretty stable. And he proposed this molecule as a basis for building structures with the DNA. So if you extend the arms of this, I don't know, we usually put this arrowhead at the 3' end. So this green strand will have 3' end here and 5' end here. And so this portion of the strand is complementary to this portion of the red strand, and this portion of the red strand is complementary to this portion of the blue strand, and so forth. And due to the nucleotide complementarity, we end up with this assembly of this structure. So if we, in some cases, extend the 5' end here is expanded, and these are free nucleotides that can join with other nucleotides, we can use this molecule now as a Lego block and build up new structures and have, start building an array of building blocks. But if we extend the arms of this molecule, then it becomes quite flexible. The DNA becomes flexible, and they can actually move around and have different mobility and different types of structures. We also, this is an example of triplex molecule, TX molecule, where you have three helical, helical duplices, right? Each one of this is a helical duplex joined together through a threading of different strands. As you can see, the yellow comes from the first one, moves through the second, goes in the third, and the blue one joins first and the second, and so forth. So this kind of molecule is also very sturdy and rigid and has been used in forming a variety of two-dimensional arrays. So these are some examples of two-dimensional of DNA self-assembling. That this image here is the first two-dimensional array that was obtained, and I think in 1998 or 1999. And these are other different possibilities that people have picked up and brought to life. So where our interest came to life, and when we started thinking about three-dimensional structures and what types of shapes occur in 3D. So I can think of self-assembly as information processing in 3D in some sense. I have every molecule can be thought of as an information unit that when they assemble with another molecule, you have some kind of information processing. And we probably need a notion of shape processing, and rather than one-dimensional computation and one-dimensional information processing that we have currently in all our computers. Everything is, in fact, transcribed through zeros and ones. When we think about molecular self-assembly, I would like to think about three-dimensional structures and some kind of shape processing. I'm not sure that we actually have a good mathematical notion for that. We're working on it, but okay, I will give you an example of how one can think about it. So the graph structures, I will talk about the graph structures in 3D. I thought I would talk about algebraic structures, but with DNA origami, but I won't have time, so I'll skip that. I'll talk about DNA crystallographic structures. All right. So let's move on into spatial graphs. So here's an example of something that an experimental, I guess, result that we got with NET some years ago, I guess a long time ago, which was a mathematical problem solving vertex colorability problem. And we know what the vertex colorability problem is. Given the graph, we want to color the graph with certain number of colors. And in our case, we are focused on tree coloring. So we want to use three colors, green, red, and blue, to color the vertices so that no two vertices that are adjacent or incident to the same edge will have the same color. So with this example is here, one coloring of the graph. So we decided that we can try to solve this problem through actual graph construction by DNA. So we designed molecules, junction molecules like this. So for example, vertex 6 here has degree 3 with 3-valent vertex, right? We designed 3 copies of 3 valent vertices like this. We extended the 3' end with 3 nucleotides of this strands, such that the portion there are 3 portions of this of these 3 nucleotides. 2 of these portions, say A and C, are encoding with nucleotides that encode precisely the vertex and the edge where this arm would belong to. And the third coding, which is B in here, the middle one, would encode the color. So, so this particular molecule here would represent a vertex 6 with color red. So I have a vertex 6 with color green and vertex 6 with color blue also in the test tube. The same with vertex 5. Vertex 5 is actually 2 valent vertex, so it will be just a single duplex. Vertex 4 has 4 valency, right? And we have a structure of this form. So these are a copy of each color per vertex is present in the test tube. So the way we designed it was that when 2 vertices join together with the same color, so the complementarity sticks together completely, then we encoded a restriction enzyme inside, and we, in particular for red, say it's hint 3, the enzyme will actually cut the structure, will cut this edge. However, if the molecules join together through the complementarities here of A and C, for example, when they join together, they have different color, then they will form a bubble. There's no complementarity between red and green, and this bubble cannot be cut with any of the 3 enzymes corresponding to red, green, and blue. So the whole process ended up like this. You design these molecules, 3 copies per vertex, right, for the 3 colors. We put everything in the test tube, wait a little for the self-assembly to take its course, and apply 3 enzymes, hint 3 and all the other 2 enzymes. The enzymes will cut all of the edges that are same color that have joined together. The only thing that remains afterwards would be something that is actually a good coloring of the graph. So this would be a molecule like this, where we have every edge will have a bubble where the enzyme didn't cut. So the process is straightforward. It was, if you think about it, once you design the molecules, it's O of 1, right? I have the same number of steps. You put everything together, apply 3 enzymes, and fetch the molecule that is that remains. However, fetch the molecule that remains turns out to be a little bit difficult, because you have to prove that the molecule that you have designed is actually formed. And that was the hardest part of the experiment. But before I go there, I want to point out what we did. So this was natural, this was just self-assembly and enzymes. That's all we had, right? So if natural self-assembly process that produce complex molecular shapes perform computation, this was a result of computing or figuring out how to color a graph with 3 colors. So what do molecules compute when they actually form different structures? And does this computation confer selective advantage? Another very interesting point was that the molecule that remained after this process is a non-minimal energy molecule because of the bubbles. There could have been lots of formations of the same graph with the same coloring of the edges, where you have a full complementarity. But because of the environment with 3 enzymes, none of them survive. So in fact, we end up with a complex shape molecule in a complex shape that was not minimal energy. molecule. So now we go into how to prove that we have the graph that we actually wanted to have. So in the design, we made the design such that when the edges come together, there will be one molecule. Well, afterwards we applied the ligase, which actually joins the nicks of the two molecules. So the phosphate and hydroxyl group are joined together, and they the strands that had a nick actually are now nickless, hopefully. So in the design, we actually made this, I don't know if you can see this purple molecule, that is a purple strand that traverses the graph such that every edge is traversed by this strand, not the whole graph, but every edge of the graph. So we actually fetched this strand, and then we just sequenced it, and we said, well, yeah, sure, this is the vertex, and this is another vertex, and this is an edge between the vertex, and so forth. Because of this strand, we were able to prove that a graph indeed formed and solved the problem. So that brought us to when you design these structures, you want to have something that will traverse the structure and show that the structure exists. So we need reporter strands. These reporter strands have the following property. I want to walk in the graph that visits every edge at least once, and at most twice. But when I visit the edge in second time, it has to go in the opposite direction, because that's how the DNA works. And I have to preserve the structure of the graph. So if I have an Eulerian graph, then the Eulerian circuit would actually give me the answer. But if I don't have an Eulerian graph, then things become a bit more complicated. So the way we prove this, we considered this problem was we took the thickened graph. So we are going to take every edge of the graph as a flat strip. And every rim of this strip will be, say, a DNA strand, but they will be oriented in the opposite direction. So a thickened graph of f of G will be a compact orientable surface to the dimensional manifold such that G is topologically embedded as a one complex in f of G as a deformation retract. And this structure, f of G, is not necessarily unique. Actually, there may be many. However, the boundary components of this thickened graph are the DNA strands that we want. So here's one graph, and these are the two possibilities of surfaces to represent the same graph. They are topologically different. This one has 3 boundary components. This one has a single boundary component. This graph has only this one. However, I changed the topology here. I will get something equivalent to this. So a reporter strand for this thickened graph is a boundary component that traverses every edge at least once. So we showed that every multigraph has a reporter strand. We can find a reporter strand. Later on, Joel Moynihan and Mark Ellingham showed that this process of getting the optimal reporter strand is NP hard. And it turned out that the problem is a variation of Chinese postman problem, which is known to be NP hard. And I mean, many constraints can be done in that way, boiled down to Chinese postman problem. And actually this year, just a couple months ago, Demain, Kirkpatrick, and Lin, Eric Demain is a professor at MIT. You might have known. So they just posted on arXiv another variation of Chinese postman problem with threading of the graph, graph threading and turn costs. So another question which might be of interest to this audience is this one here. How many distinct nodes does the boundary component of FG corresponding to the reporter strand yield? And we are still, this is a question we have not been able to answer. So a little bit about how the proof went. I mean, it was not that difficult. Once you start thinking about it, we have if I have a three junction molecule like this, if we introduce a single half turn on one of these, I add an extra 5 nucleotides on one side. Then this green, instead of green strand that goes this way, instead of going to the right, it starts going to up. And the purple, instead of going up, starts going down. So we change the connection at the vertex of the boundary components. And given this graph, for example, these are two possibilities. This is a planar graph, so I can actually have this planar representation and look at the boundary components. There are 5 of them. Or if I change the connection at 2 vertices here, the boundary components are brought down to a single boundary component. So this graph here is traversed by, I mean, the thickened graph has only one boundary component. It all depends on the embedding. So if I want the best reporter strand for this graph, I don't have to traverse this cycle and this cycle twice. Then I can embed the graph in a single torus. But if I want a boundary component that traverses every edge twice, then my embedding is actually in a double torus. So it turns out that the number of boundary components depends directly to the, well, of course, depends on the genus of the Euler characteristic and the maximum and the minimum genus of the surface where this graph is being embedded, cellular embedding only. So, and actually it also goes by parity, right? This number can be either all evens or all odds. The change of the number of boundary components. Okay, I go into a new topic now about DNA origami. I don't know if there are any questions.
Actually, yes, Natasha, you mentioned energy. What kind of energy you think of?
Oh, the temperature. Higher temperature, molecules move faster, lower temperature move slower. Of course, so the whole self-assembly process actually in this DNA self-assembly processes happens when you cool warm the solution to certain degrees, usually about 40-50 degrees, so that the junction molecules don't fall apart. And you cool them down slowly, so they move around, but as soon as they find their complements, you try to fix them so they don't start falling apart. You cool them down slowly up to 4 degrees. And
okay, so minimal energy states.
Oh, minimal energy state means, okay, that there are still potential of my, yes, I should have mentioned. Minimal energy means that there are still unbonded nucleotides that can perform some kind of interaction with other molecules.
Oh, I see. Okay, thank you.
Yeah, Natasha, I had a question about that experiment where you did the computation. Did you do a gel electrophoresis? Could you see difference in the products?
Yes.
In the gel?
Yes, yes, we did gel electrophoresis, of course, and the highest band was the molecules that we were looking for, of course, but then we did the 2D gel electrophoresis, and we discovered that there are actually two embeddings of the molecules. So there are two topological embeddings. We also did the topo analysis. So the one that I showed you in my slide was one of them, but there's another topology that we discovered of the same strand, same structure. This topology, a different embedding. So we could identify two topologies of this molecule.
Interesting, yeah.
Okay, so any other questions before I move? Okay, I want to say a couple of words about DNA origami and reporter strands. So DNA origami were introduced by Paul Rotemund, and how DNA origami works. There's a cyclic molecule. Well, this is a plasmid, so there's something that's not used here in this structure, but it's a long molecule threading the structure throughout that is being joined together with multitude of short molecules that join different parts of the molecule together, and then the formation happens. So again, you start with everything and some temperature and slowly cooling down. When they cool down, the different complementary parts of the molecules come together, and these small molecules that are called staple strands, like stapling this part with this part, slowly bring this structure together. So this was the paper ended up as a top, front page of Nature, 2006, and there was many structures that were made. The smiley face being one of them. He made close to 3 billion of this smiley faces. Very happy. Nature. But here's an example of the design. So U is a design and V is the actual experimental result. So it turned out that DNA origami works very well for a variety of structures. And the field just exploded. They started building a whole bunch of structures in different, so this is a, two-layer two-dimensional array. They started building three-dimensional vases and things. Then some aptamers that would open up in cell environment potentially and things like that. And now we are talking about precise molecular arrangements applied on the DNA origami. There are about, I don't know, 300 to 400, I guess, labs currently using DNA origami for a variety of purposes. I want to go back to one for this audience might be interesting design, which is the DNA mesh. And if you have done any computer graphics, then you know about this bunny. And what the Benson lab did, actually in Karolinska Institute in Sweden, they designed variety of shapes with meshes, and they with DNA origami. So these are three-dimensional, like hollow things, right? They're all, in some sense, sitting on a ball. So the way it was done, and this was done by Malik Mohammed, who ended up in our group as a postdoc. You take a target shape, make a triangulation, and when you make a triangulation, you want to make it such that this is Eulerian, because as I mentioned, when you have an Eulerian graph, it's very easy to find an Eulerian circuit that would traverse the whole structure. So you identify edges where you have to double the edges to make it Eulerian, like this. Once you have the Eulerian graph, you design the staple strands, and the staple strands would join different shape of the structure. The Eulerian circuit would be the origami traversing the structure. So these were known as A trails in Eulerian graph. How do we actually traverse a graph? Well, if I have a vertex of 8 degrees, I can traverse around, go through the vertex first, join 2 edges, then another 2 edges and another 2 edges. And in some sense, we are pairing edges traced at each visit. If I do pair the neighboring edges, then I work on a trail, or I can look at non-intersecting pairing, because both A trails and non-intersecting pairing are sure to provide me with unknotted routing whenever I have a mesh on a ball, topological ball. Well, the point is you start with the unknotted origami, big cyclic molecule. So whatever we are going to traverse with this molecule, it better ends up being unknotted. So the question ended up with what happens if I have meshes on other types of surfaces? And this was a joint work also with Masahiko Saito. Well, if you think about it, if I have a K7, which is a full graph of 7 vertices, Conway showed a long time ago, and with Gordon, that any embedding of K7 in R3 produces at least one cycle that is being knotted. So I cannot put K7 somewhere and produce an unknotted shape. However, if I embed K7 on a torus, it would look something like this. So the 3 cycles that I have identified here are red, green, and yellow. Purple, green, and yellow. So the purple goes like this, and the yellow moves around, and the green also. The interesting thing is that I can do a checkerboard coloring. And once we do a checkerboard coloring on this embedding, we can produce an A trail. And this A trail now makes sure that we have unknotted structure. So this embedding on the torus of K7 is producing an unknotted, I guess, traversal of the graph. So we can prove now a couple of other observations. If I have all non-intersecting scaffold routings are unknotted for checkerboard colorable graph on a torus, and there always exists an unknotted non-intersecting scaffolding for any checkerboard colorable graph which is embedded in a close orientable surface. So the important part is checkerboard colorable. And well, it depends on the genus, of course. If I have a torus, then everything is unknotted. If I have other orientable surfaces, I can always find one that is unknotted. I went back and talked to Ned, and Ned told me, well, I tried knotted scaffolding in my lab and I produced different structures. What can I do with the knotted? If I can actually took this viral plasmid and knotted the viral plasmid and wanted to know what kind of origami structures I can get when I have a knotted structure. And then we observed different things. If I have an Eulerian graph, then I can actually have a knotted routing of the graph when this graph is embedded in a standard torus, only if I have a non-checkerboard coloring embedding. And for every genus, actually, n greater or equal to, we can find infinitely many Eulerian graphs that have, in fact, checkerboard colorable cellular embedding, but they're all knotted. So this was an interesting observation. There are lots of open questions that remained from this work. We are still working on it, but I just wanted to point out that a small question from the self-assembly turned out to be mathematically interesting. And I have about 10 minutes maybe to finish with the DNA crystals. In January 2023, I just called a bunch of people. We were talking about DNA crystals and topology of DNA crystals. So this is the gathering. So you can see here Lou Kaufman. So Wout from Amsterdam came, Eleni Panagiotou, some of my postdocs here, and Simon from Ned's lab was here. So the DNA crystals are fairly first time made out of this tensegrity triangle. You have 3 duplexes. They're joined like this. Actually, I have a physical object here that is a 3D printed object. And they join together to form a crystal. And the crystals are actually quite large. These are 200 micrometers. Some of them were so large that they were visible by naked eye. So this is the image of the DNA crystal. So I want to point out, I don't know if you can see, there are 3 helices, 1 in every direction. So let me go back and can you see my screen now?
Yes.
It's black. It doesn't have anything in it.
Yes, I will get it. So first I'm gonna show you the standard 3D structure. The molecule that made the first crystal. This is 2T7. So this is how it is. And I'm identifying the circles, the cyclic strand in the middle, joining all 3 duplexes. But as you can see, there are 3 helices that are going in 3 different directions. If I take a look at the crystal, then it looks something like this, where I have also circled the central cyclic molecule. And however we put it, we end up with 3, 1 helix goes back and front. So this is 3T7. The main part is that there are 7 nucleotides between one junction to the other junction. So let me do 2T3. So this was also obtained in the lab recently. So here's the cyclic strand in there that doesn't go outside the center of the molecule. Actually, the cavity in the center of this molecule is so small that you cannot even put a water molecule inside. But there are only 3 nucleotides from one junction to another. So from one junction to another, there are only 3 nucleotides. It also forms a structure, a crystallographic structure, as you can see in 3D. And what I want to now, let me see if I can, oh, I should have moved it. Let me see if I can move this away. I should have moved it further, sorry. I want to put them together. Let me see if I can put this together. No, they're only together, sorry. Well, one of them is with 3 nucleotides and the other one is with 7 nucleotides. I want to, maybe we can just look at this, put it in one side. So I'm gonna focus on this duplex, which goes on top of this duplex. So I want to call this a positive crossing. And if I take the 3 nucleotides, the same duplex goes below this duplex here, which would be a negative duplex. So one is left-handed, the other one is right-handed. So it turns out that there's a chirality between these 2 molecules, the different chirality. But when I look at the 5 nucleotides between the junctions, well, I cannot even get a triangle. With 5 nucleotides, I just don't have a triangle. And However, I do have a crystal. I can form a crystal, and the crystal turns out to be more or less two-dimensional. I have one going this way, one goes straight back and forth, but there's nothing going in the 3-dimension. There's no duplex that goes in the 3rd dimension. So the question was, what makes one triangle left-handed, what makes one triangle right-handed, and what makes this weird shape where the central strand doesn't even make a cyclic molecule and I don't have a triangle? So we went back and started looking at, let me see, I want to go back to my talk. You see my talk?
Yes.
Okay. So I will skip this. This is a joint work with Ned's lab. Simon was the person we talked the most, and Van is our postdoc, and also with Masahiko. So we looked at the number of nucleotides between junction molecules, and for what number we actually end up with a triangle. What are the necessary conditions to get this triangle formed? And when do we have left-handed, when do we have right-handed? How about the chirality? So we started modeling this cyclic molecule in the center and with 3 curves, one being on each side of the triangle. And let me see, so this would be a curve. We also tried to model the helicity, which is 3.4 nucleotides per 1 helical turn, 2 nanometers of the diameter of the DNA, and we use 10.1 base pairs per helical turn, actually. This was important because most of the helical turns were actually, the DNA was a little bit tighter than it is standard. So we modeled the helix such that at the end we moved it around. I'm running out of time, so I just want to point out that we took the helix so that it is exactly lies from the beginning to the end on the x-axis. And then we started rotating this helix around this x-axis and checked what kind of curve we get when you look at all 3 sides of the triangle. So this would be one rotation if we rotate this for one side, or this would be another rotation of that curve. And this represents the central cyclic molecule of the triangle. Of course, we looked at the complementary strand as well. We looked at the smoothness, actually how smooth the connection between the 2 curves are. We looked at the triangle cavity, what happens in the middle, how far the molecules are in the center. And we looked at the cylinder tilt, the duplex, actually, how tilted the cylinders are. And these are the curves that represent those measurements. So this is for n equal 3, and this is for n equal 7. So the highest tilt with positive cavity and actually the very sharp smoothness is in here. And for n equals 7, we have the highest tilt in the opposite direction with positive cavity, but very smooth connection. So those are actually the mathematical properties that we have to have in order to get the triangle. I have to have the tilt. We have to have either local maximum or local minimum and the maximum cavity inside. We also looked at the complementary strands. They had to be a certain part aside because this was the lowest measured cavity that we distance between the complementary strand experimentally. So here it is. If I am in this location, I have this jam of the cylinders so each cylinder corresponds to one duplex. of the triangle. But if I'm here, then we end up with this configuration. Same here for n equals 7. If I'm on this location, we have these nice triangles, cylinders. But if I am on this location, they're all jamming together. So we went and we identified all of this, and we said yes, no, no. So whether the triangle forms, we could actually confirm mathematically, and we could also confirm left-handed versus right-handed chirality. And this is, I think this shows the right-handed versus left-handed. So here we have left-handed crossing. I guess you see about how the right is on top of this curve is on top of that curve, and here it should be opposite. This curve will be on top of this curve. And these are the results. The paper appeared at Proceedings of National Academy of Sciences in this summer. But these are the results for different different distances between the junctions. So these are mathematical predictions, and at the right, we actually have superimposed the crystallographic structure from the lab on our mathematical curve that we computed as the best solution for the triangle. Of course, for n equal 5, we could not get anything. And these are the experimental results of the crystallographic structures. I just want to point out that mathematically, there's lots of questions about characterizing all these different crystallographic structures. They're all, and the periodicities of the strands that are traversing these structures. And we are trying to find invariants that can distinguish these building blocks, different building blocks. And we want to characterize the strand topologies that can appear in these structures. Recently, well, out of this joint, when I invited a bunch of people last year, well, Masahiko, Mohammed, and Wout Molwakker from Amsterdam ended up with a paper where they wrote the noted Quandale invariant for the as a starting point as an invariant for this notoid structures, because they're all open curves, right? So these are all open curves structures in the disintegrated triangle. So I just want to point out that the lab has come up with, we have many different structures to work with and to consider and to look at the shapes, and mathematically want to characterize what can appear, what cannot appear, how to distinguish one structure versus another. And I just want to finish with this book. And I'm not advertising a book that this is a joint book with Eric Minfrey from Caltech. We have actually negotiated with Springer that this book is open source, so you can download it and look at it, but there's zillions of questions there and up-to-date DNA nanotechnology. Thank you very much. I am sorry it took me a little bit longer.
Well, Natasha, it was a great talk. So now we're open for questions.
Well, from the advert of the book, DNA Visions or Visions of DNA for the Future, I wondered, can you just comment very briefly? I imagine there are many, many important applications in the future coming, but would you give me an idea of the possible application for DNA origami and, well, crystals, I guess, but origami is an interesting thing from a mathematical viewpoint. Can you give me an idea of applications?
So most of the time people are using origami, well, either as a container to store other molecules, and being a DNA, we hope it could work in natural cell environment, or because we can actually access different, very precisely different parts of the DNA origami. So DNA origami is used as a scaffolding for other nanomaterials, in particular metal organizations and nanowires.
I see. And if I may ask one further question is any of this work related to possible implementation in DNA computation?
Well, DNA computation, from my point of view, is something, well, that I originally started thinking about, but now I'm looking at this as just being information processing, and how to use the notion of computation and transferring of information between one molecule to another to, for example, do data storage, or do encoding, or design material that you want to design. So yes, it is DNA computation, but not in this way of computation as we are used to with our electronic computers. I don't think this can compete with that. Okay, but it can compete in ways that our computers don't.
Very clear. Yeah, sure. Different kind of scale, so to speak. Yes. Thank you.
Natasha, I had a question about K7 that you embedded on the torus, and somehow
the checkerboard coloring. Yes.
Yeah, somehow you got the coloring scheme, and you got a reporter curve that was unknotted. Now, this reporter curve is going to be like some sort of companion of K7 itself.
Right.
Certainly with K7, there's a lot of knots in there. You know, the orphan variant doesn't go away, but
right, this one.
Where did the knottedness go?
Well, the embedding is such that you can, once you do checkerboard coloring, that's it, because now you do traverse it. I'm not claiming that we have found one traversing that is unknotted. Okay. If I follow the yellow by itself, right, which would not be, so if I follow the yellow here and yellow here, I would cross this vertex. So I wouldn't follow the checkerboard coloring. I will cross the vertex, go across it. So if I follow the yellow, then you end up with the torus knot, right?
Yeah.
So the knotting is still there, but the point is that you can find one traversing that is unknotted for the whole thing.
Got to be real careful when you do it. Yeah.
So that was the point. But both green and the yellow cycle are actually torus knots here.
Yeah.
The green goes around and the yellow goes around, and they're both torus knots. Yes. But if I follow the yellow, I go across the vertex, and the green across the vertex. So that's not good. I should go yellow, then green. And that would be good. In some sense. So we in some sense, we avoided the knottedness, but only for one traversal of the graph.
Okay. Yes. I had another question about chirality. In those crystal structures, you're able to define chirality by looking at kind of crossover patterns at the vertices somehow.
Yes.
So did it twist in a right hand or left hand way?
Now, right. This crossings here of the cylinders.
Yeah.
And it turned out to be completely different. I mean, let me see. So here's a 7 and here's a 3. I mean, I hope you can see that chirality is opposite.
Yeah, okay. And there were some structures where there was no chirality at all. So
No, you could not. There are structures where it's impossible to get this triangle. It just doesn't form.
okay.
And that means that this three curves don't line up anywhere the way that they're supposed to line up. And it's funny because, you know, so if I have for three, you can imagine, I mean, three is just three nucleotides. It's so tiny, but still you end up with a triangle. But 4, 5, 6, no.
No.
Yeah. 7 and 8, yes, but it's right-handed, not left-handed like 3. Then 9, 10, 11, no, nothing happens. 12 through 14, left-handed. 15, nothing happens. And, you know,
Interesting.
yeah, it was very interesting. But different chirality happened to appear with these triangles. Somehow in the first, if I look at the 10, if I look at the chunks of 10 in the nucleus between the two junctions, say 10, 20, 30. So if I'm in the first third, like 3, 4, 5, or 2, 3, 4, I'm sorry, 5 usually doesn't work, but 3, 4 or 2, 3 works. So 3, 12, 13, 14, they work. They're left-handed. 5 is in this, you know, transitional form when nothing happens, and then you have 16, 17, 18, or 6, 7, 8, or 7, 8, where you end up with right-handed. But then again, around 10, 9, 11, maybe not, but nothing happens. That's a transition. And then you move on into the next 10, start with left-handed. So it's go left-handed, nothing, right-handed, nothing, left-handed, nothing, right? Interesting.
Yeah, indeed. Okay. Are there any other questions?
Yes, Natasha. About the first part of your talk, have you thought about ribbon graph polynomials associated to the ribbon graphs that you have talked about?
We have looked at polynomials, but not ribbon graph polynomials, actually. So where I am. No, we have not associated polynomials with this, but that might be interesting. Well, I guess the main question that we still have, how to, maybe would those polynomials be related to these questions of how many distinct knots a boundary component will actually yield in the reporter strand?
Yeah. What I mean is that after that you have this ribbon graph in your embedded in your surface, you can associate many different polynomials.
Yes.
Like the ribbon polynomial, or like the monochromatic, or
Exactly.
Right. But you have never associated these polynomials to your real ribbon surfaces?
No, we have not.
Okay. Just to, I mean, you know how to study these ribbon surfaces, maybe to use the polynomial associated to that, no?
Yeah, sure. That might be a nice avenue to take, in fact. We have not thought about it for some reason. I'm surprised that we haven't. Thank you.
Thank you.
Thank you.
Well, thank you. Great talk.
Thank you very much. And thank you for the invitation.